Hey guys, this is Lei. Welcome back. In the previous video, I showed you how to perform differential expression analysis. In this video, I'm gonna show you how to perform pathway enrichment analysis and how to visualize the enriched pathways. The most popular pathway enrichment plots include bar plots, dot plots, and GSEA plots. There are also other options such as ridge plots, networks, offset plots, and heat maps. The most commonly used pathway databases include Gene Ontology database and KEGG database. The Gene Ontology database consists of three components: the biological process, cellular component, and molecular function. Go terms in the Go database are stored as directed acyclic graphs, while genes in KEGG database are stored as networks. Both databases can be found in the molecular signature database, which is a collection of basically all pathway databases, and classified them into different categories. For example, we have hallmark gene sets, we have ontology gene sets, we have KEGG gene sets in C2 category, we have onc gene sets, and so on. There are two ways to perform pathway analysis: the overrepresentation analysis, which tests if your DEGs are significantly overlapped with the pathway genes, and the gene set enrichment analysis, which tests which genes are contributed to which pathways. The statistical method used in the ORA is usually Fisher's exact test, which calculates the p-value for the overlapped portion. Well, the GSEA is a bit complicated. It ranks all the genes in your dataset first. For D analysis, usually we use log fold change for the ranking. For other cases, you can use correlation with the phenotypes. It then walks down the ranked gene list and increase the score when the gene is in the pathway gene set, and reduces the score when the gene is not in the pathway gene set. The total score is represented by the peak in this plot, and the genes contribute to this peak are known as the leading edge subset. For ORA analysis, we only need one gene list, nothing more, but for GSEA, we need the whole dataset. Both analyses can be done in R. As usual, our first step is to install the packages. Among them, clusterProfiler is the core package for the enrichment analysis. We also need some genome annotation the packages. For example, for mouse dataset, HS for human, RN for rats. The pound sign in the beginning tells R not to execute the code. As I mentioned before, as a data scientist, our first step is always to prepare our data. So for ORA, we need to prepare our DEGs. So we're reading our DEG table, set up the threshold, and subset our DEG list. So we use the function called enrichGo. It accepts a list of genes. So for ORA, we usually separate the upregulated genes and the downregulated genes. And this code is to make a gene list for upregulated genes and downregulated genes. The format is like this. So once you have the gene list, feed it to the enrichGo function and set up other parameters. So on equals all means we want all three components of the ontology database. The function resulted in enrichment object. In the object, we have an enrichment table. So we extract the table and save it to our computer. The table has this format. We have enriched Go terms, description, gene ratio. So 13 means the overlap genes. 127 means the DEG we have, the number of DEG we have. So we have the adjusted p-value and the q-value. So filter your enriched pathways using these values, ignore the p-value. And the count means the number of overlapped genes. You can also show the top enriched pathways in a dot plot using the dot plot function. So the input of the function is the enrichment object we just generated in the previous slide. So the object, the result is a ggplot object, so we can save it using the ggsave function. For GSEA, it requires a ranked gene list with a format like this. The numbers are the log4 change. The name of numbers are the genes. So this is a named vector. To make a named vector, we use this code. So ranked log4 change first and name the log4 change. Once we have the list, we feed it to the GSE go function and set up the other parameters in the same way we did for the ORA analysis. And then we extract the resulted table, the enrichment table, in the same way, which has this format, the similar format. We have go IDs, descriptions, and we have normalized enrichment score, which we have to pay attention to. A positive NES means the goal term is enriched by the top ranked genes, which are usually the upregulated genes. A negative NES means the goal term is enriched by the bottom ranked genes, which are usually downregulated genes. So we can filter the enriched pathways in the same way with the p-value and the q-value. The core enrichment columns, the column represent the leading edge genes. You can also visualize the top enriched pathways in the dot plot in the same way. But for GSEA, we get a chance to plot the GSEA plot for individual goal terms. So for this, we first specify which goal term you want to plot. So where to find the goal term? It's in our previous table, enrichment table here. So find your favorite goal term to plot and feed it into the GSEA plot function. So the input is also our enrichment object generated in the previous slide, and specify which goal ID you want to plot and save the plot to your computer with the ggsave function. There are also functions specifically for KEGG enrichment. So for example, ORA, we use enrich KEGG, and GSEA method, we use GSE KEGG. So how about other pathway databases? Well, there's a way you can query your DEGs against all the pathway databases. So first, we download the MSeqDB, the whole MSeqDB database. You can find the link in my website. Download the whole database, read it in, and use the enricher function for ORA analysis. And the input is your gene list, and the other parameter is the MSeqDB you just read in. And you can get the enrichment table in the same way and save it to your computer. For GSEA, do the same thing. Read the whole MSeqDB database and call the GSEA function. And the input, of course, it's a named log fold change vector. And the database is MSeqDB, and extract enrichment table in the same way. This is all I've talked today to do the enrichment analysis in two ways in multiple pathway databases. And you can go to my website and copy the code, try it on your own. in the next video, I'm going to show you how to predict cancer subtypes using RNA-seq data.